I'll just stay down here and just like it happens with the computer. Yeah.
Did you just look for Twitter? Did you look? I just said it.
Oh my gosh, there's 4 viewers.
It's warmer than.
I'm going to give it a shout out. All right. That's awesome. I'm not surprised. Yeah, you can just schedule it on marketing. I have other. That's fine. Whatever works.
Oh, yeah. No, I'm taking pictures.
Not very high.
Yeah.
Oh, good.
Richard, apparently this guy is a new SSRP student, so you guys should be friends. Except, I think his name is Vlad. He just introduced himself. Anyway.
Also, you have something shiny on your nose. There you go.
You know all these people?
Mostly. Is there anybody there? Yes. Hi. Oh, there are 7 people. Hello, 7 people.
Susie, Betty, Donna.
Some friends from undergrad.
Yeah.
Hi, this is Richard, and he did the SSRP with me.
Oh, hi. SSRP, is that what the fellowship that she's doing?
Oh, that was what she did over the summer.
Oh, okay.
I forget what it stands for. Yeah, but that was when she first came here and fell in love.
Oh.
And so she stayed really in touch with the network. Yeah. Well, she's always in touch with lots of networks. Oh, Stanford Summer.
Oh, okay.
That would make sense.
That does make sense. Your family is the bookends. Bookends. Yeah, 2 over here and 2 over here, and then all the stands are in the middle.
No, they are. They're all because you can still show up for the food. And also, there's I bet it'll be after. Absolutely.
Oh, I thought you were getting a picture of us.
No, I could do that too.
Oh, here, let me see. Hello. Okay. Have you done this before?
No,
You gotta find the horizon line.
and you just go.
Yeah, you just follow it. So
And then
you start at the side. The arrow says start. There you go. Nope, just click the. There you go. Yeah, turn the sideways off.
I don't want to keep an arrow on the line. It's not like it does.
You're the first MVP sheet.
Don't remember how many. I bet someone was starting or not starting. And I'm not sure, maybe the might be last or She might be here as well. She's the so she might be here just they've all gone. Always happy. I think every single profession there's been one committee member. Last time I went to like calling, running around. No, Trisha's the first from our year. I'm gonna cry at this. Trisha's the first from our year. So I'm her taxi. Maybe not. I met her sister. No, I didn't even know it was happening. Because this is usually more of a big deal.
Yeah, that's what no.
Oh,
There it is. There it is.
So welcome to the Thesis defense of Trisha's name. It's my great pleasure.
The microphone's not on.
Not on. Okay. It's my real pleasure to host this event as the head of the lab, or Trisha's direct mentor. My role at this event is really just to either embarrass myself or embarrass Trisha, and so I will try to do both. So I brought a special for the occasion. So this is the thesis defense on systemic immunity and neurodegeneration. These are the twists and turns of becoming a doctor, but also the story. Once upon a time, in a lab far away, studied a girl who did not quite know what she wanted to be. And so when Trisha came to my lab, she went through various ups and downs, as this is frequent for graduate students, as we came to learn. And so one time we had a discussion whether she wanted to be an architect. I suggested she should consider this and stick with science. She also dabbled quite proficiently in arts. So these are paintings from Trisha that she made for a thesis defense of Jing, who defended a couple of years ago. And you can see one of the paintings here, and then this collage here that I showed you before. So she's a very talented artist and painter. Sometimes the paint gets on her, so the colors are not. She has many other talents. She's also a tailor, so she made this dress herself. She also learned to be in the center. Maybe she was asked before. And again, this time horizontally. She also considered to be an athlete or a model with escort. And of course, everybody knows her cheerleading skills. This is really bad in colors, I apologize. So here she's cheerleading herself. Most of the pictures you try to take from Trisha, she will jump up. She will also instruct others to do the same. This will be fascinating. And note her tiara here. So they actually jumped on, is it 6?
Yes.
It's 6, okay. It turned out games did jump higher. And 4 years later, after lots of cheerleading, this is me here. And miraculously, the princess, notably the Saitov princess. So she will talk to you about these various butterflies here and the cells in this picture. And when I showed this to my wife Christina, we just have a look at it. The first thing she noticed, of course, so this is a royal celebration for Princess Trisha, and after the thanks, we will have a reception in the East Courtyard.
I hope that's recorded.
That was sufficiently embarrassing. All right. You hear this? All right. Hello, everybody. Thank you for coming to my thesis defense. I'm very embarrassed now, but it's really wonderful to see so many people here. As Tony mentioned, I am a big fan of art and music, and I like to dabble in both of them. And so I thought I'd start my talk with a story about an artist and a musician. The artist is Dr. Anne Adams, who was a cell biologist in Vancouver who studied cancer biology like some of you here today. At the age of 46, she suddenly decided to change careers and become a painter. And the first things she started painting were these beautiful portraits of houses. And Anne would get obsessed with a theme and create dozens of variations on that theme, including over 27 different paintings of strawberries. Eventually, as her career progressed, Anne Adams started to paint. She became obsessed with more stark, repeating geometric forms, such as this painting, which represents pi, and each of the digits of pi are represented by a unique color. And this painting, which represents the aura from a migraine. At the age of 53, Dr. Adams painted what is considered her most famous painting, which is called Unraveling Bolero. And for those of you who could hear the music when it came in, we were playing Ravel's Bolero. And this painting is a visual representation of that song. Each bar of the music is represented in its own box. And Dr. Adams was completely obsessed with this song, and she would listen to it on repeat. Shortly after completing Unraveling Bolero, Dr. Adams experienced an unraveling of her own cognitive abilities. She became unable to remember specific words, and eventually she was unable to speak at all. Similarly, Maurice Ravel, the composer of Bolero, started his career composing prolifically very flowery, impressionistic music. And at the age of 53, he composed Bolero, which, if you have heard it, is a very repetitive, staccato song that repeats the same melody again and again. Shortly after composing Bolero, Ravel experienced his own unraveling in an identical manner to Dr. Adams. He began to lose his ability to recall words and then to speak. It is thought that Ravel and Dr. Adams were suffering from the same underlying neuropathology that robbed them of their ability to speak, but also informed their creative outputs. And that disease is frontotemporal dementia, which will be the focus of my talk today. Frontotemporal dementia, or FTD, is a deterioration of the frontal and temporal lobes. And I'm sorry, I apologize for the projection here, but this is an MRI of a normal patient. You can see a healthy brain. And if you can discern it, there's a severe atrophy of the frontal and temporal lobes in this FTD patient. Frontotemporal dementia is the most common early onset dementia, and it accounts for about 10% of all dementia cases. It is estimated to affect around 50,000 Americans, and the course of the disease is very rapid. The average deterioration from diagnosis to death is about 8 years, but the disease can last anywhere from 2 to 20 years. And both Maurice Ravel and Dr. Ann Adams died of this disease. And many people are continuing to suffer from this disease, and there is still no cure or effective treatment. So the goal of my thesis work was to try to find more information about the pathology of frontotemporal dementia that might help us to lead to better therapeutic targets. What we know about frontotemporal dementia is that there are both genetic and sporadic cases, and the genetic cases are caused by one of several known mutations, and these patients have a family history of neurodegenerative disease, and many of them have a known mutation. We've been able to identify several in the last decade. However, over 60% of the cases of frontotemporal dementia are sporadic, which means that they don't have a family history of disease and they have no mutation. For my work, I chose to focus on finding similarities between a mutation carrier frontotemporal dementia and the sporadic disease that have common pathology. So there are sporadic cases and mutation cases that both result in the same pathology, which is in a protein called TDP43, which aggregates in the neurons outside of the nucleus, as you can see here. And the benefit of choosing to study these two patient populations is that we can identify in the clinic, so before death or autopsy, the patients that are very likely to have TDP43 pathology. So the semantic dementia patients are identifiable by their clinical presentation, and the progranulin mutation carriers are identifiable because they have a family history of the disease, and we can assay their mutation. The semantic dementia clinical presentation is the semantic variant, and this is the same variant that Maurice Ravel and Ann Adams had. And it is called primary progressive aphasia, which means they lose the ability to speak and recall words in a progressive manner. So if you presented a patient with a set of items on a desk and you asked them to pass you the stapler, they might not be able to do it. Not because they don't know what the object of the stapler is, but they can no longer connect the word stapler to the object. But if you handed them the scissors and asked them to what they're used for, they would be able to properly use them to cut paper. And this variant always presents with a TDP43 pathology underlying it. The progranulin mutation is one of the more common mutations that will result in frontotemporal dementia, and it's a very interesting one. So progranulin, and the structure is shown here, is a gene that encodes a protein that is made up of many discrete units that can be cleaved that have different functions. Interestingly enough, the progranulin mutation in the patients leads to haploinsufficiency of the progranulin gene, which means that the patients have only one working copy of progranulin and the other copy is not expressed. And it's actually been found that the progranulin mutation carriers have less than half of the normal levels of progranulin. So here you can see control individuals and frontotemporal dementia patients without a progranulin mutation have normal levels of progranulin, whereas the progranulin mutation carriers have less than half of the expected amount. Progranulin is a very interesting molecule to an immunologist because it is most highly expressed in the monocyte lineage, macrophages and monocytes, which are very important immune cells. And it's known to act as a growth factor, and it's important in wound healing after an injury. It is also thought to be an anti-TNF molecule. It was reported to bind to the TNF-alpha receptor and act as an antagonist, although this has been a little controversial if it actually binds to the receptor, but it certainly seems to decrease TNF-alpha signaling. Importantly, we also have a mouse model of the progranulin mutation carriers, and similar to the humans, we can knock out progranulin, and we see cognitive deficits in the mice, including in social behavior and in learning and memory. So we have several routes to the same underlying TDP43 pathology with the sporadic frontotemporal dementia patients, the progranulin mutation carriers, and our progranulin mouse model. And I hoped to use these three different routes to the disease to find out what they have in common that might tell us something about the underlying pathology of frontotemporal dementia. However, the brain, where the real pathology happens, is a little bit difficult to access, and so I hope to find a way to learn more about the disease without having to get autopsy samples. So I took advantage of the fact that the brain does not exist in isolation. It exists in the context of the body, and it is connected to the body via the systemic environment and the circulatory system. And we know that changes in the systemic environment influence the brain. Some of you might have heard in the news recently, there was a paper from our lab in Sal de Leyda that found that factors in the young systemic environment can rejuvenate an old brain. So we know that there's interaction there. And we also know that the brain communicates with the systemic environment through the blood by secreting molecules. So we hope to see if there were changes in the central nervous system that were reflected in the blood, or changes in the, and to see if the changes in the blood can affect the central nervous system. So in the blood, the two interesting parts to assay are the plasma and the white blood cells. So first, we looked at the plasma. And we wanted to look at the proteins in the plasma to see if there were changes in the level of proteins expressed in the plasma of the patients compared to age-matched control. To do this, we used a technique that was developed in the lab called a plasma protein array, and it's an antibody array that works in a similar way to a DNA microarray, but instead of using DNA probes on a glass slide, the slides are plated with antibody probes in discrete clusters. And then the protein, or the plasma, is incubated on the slide, and the proteins will bind to their antibodies, the specific antibodies, and then we can detect these antibodies with a chemiluminescent detection to assay the relative levels of the proteins. And the proteins that we chose to put on the array are basically any secreted soluble protein or receptor that we could get a good antibody for. And I want to say thank you especially to Marcus Rixey, Hudson, Daniela, and Jaden, who are the former and current array technicians in the lab, and they've done a wonderful job of optimizing this technology. Anyway, we were able to assay a huge number of cytokines, chemokines, growth factors, and secreted receptors. Right now, the array has over 600 different proteins on it. And this array has been used in the past to look at neurodegenerative diseases such as Alzheimer's disease, where we found a potential biomarker in the plasma. So for our first study, we took a group of frontotemporal dementia patients with the semantic dementia variety, the sporadic cases, and we compared them to age-matched controls. So we collected their plasma and ran them on the antibody arrays. And again, this is able to detect the relative levels of proteins. And the first question we sought to answer with this is, can we use the proteins in the blood to distinguish the patients from the controls? And the way that I chose to look at this at first was just to use a heat map and use unsupervised clustering to see if the top 25 factors by fold change can separate controls from patients. And what we found was that, so this is the heat map, the top 25 factors up here, and you can see that the control samples cluster mostly on this side, and the frontotemporal dementia patients cluster over here. And here you can see that the FTD patients have lower relative expression of these proteins that are largely involved in angiogenesis, and they have higher expression of these proteins that are mostly involved in angiogenesis and in inflammation. It is important to note that there's a really heterogeneous population, and we weren't able to discern a single biomarker or even a very small number of biomarkers, but we hope to use this information to find pathways that were altered in the patients that might tell us more about how this disease works. So we used ingenuity protein pathway analysis, which is a tool that connects proteins based on literature reported associations of the proteins into pathways, so we can tell if we have a list of proteins, how they are related. What we found were two highly significant pathways that were altered in frontotemporal dementia. The first is involved in inflammation, and it includes the IL-12/23 signaling axis and the TNF-alpha family proteins. So here you can see that the TNF-alpha family proteins are upregulated in frontotemporal dementia patients, whereas the IL-12/23 axis ligands are mostly downregulated in the plasma of the FTD patients, whereas some of the receptors are upregulated. And this indicates that there is an overall deregulation of this inflammatory signaling pathway. The second pathway that I wanted to mention is related to angiogenesis-related proteins, including an upregulation of vascular endothelial growth factor-related molecules and a decrease in bone morphogenetic protein-related molecules. Next, we wanted to know if the changes in frontotemporal dementia patients' plasma that we observed were similar to any other diseases, and this might tell us how the pathways are related to each other and to disease. So we used a tool called functional disease ontology, which, similar to ingenuity pathway analysis, takes literature-reported associations between proteins and this time between disease and creates a map of related symptoms and diseases. So we found that the top 25 changed proteins in our frontotemporal dementia patients are similar to changes seen in diseases such as rheumatoid arthritis, dermatitis, and asthma, which are all inflammation-related diseases and autoimmune diseases, and to endometriosis and cancer, which are related to angiogenesis-related diseases. I also wanted to point out that the IL-12/23 signaling axis is important in autoimmune disease. Here I have listed the autoimmune diseases that are associated with changes in IL-12, including celiac disease, psoriasis, lupus, and Sjogren's, and with IL-23, including ankylosing spondylosis and psoriasis. So next we wanted to see if the changes that we observed in our first cohort of frontotemporal dementia patients were, if we were able to validate that in a second cohort, as well as if we see similar changes in the progranulin mutation carriers. So we collected a novel cohort of sporadic frontotemporal dementia patients and a cohort of progranulin mutation patients, and we ran the same protein antibody array and assayed the levels of proteins in the plasma. And what we found was that there were similar protein networks that were altered in the 3 different cohorts. So here is an expanded list of the protein networks that were significantly changed according to ingenuity pathway analysis in the original frontotemporal dementia sporadic disease cohort. And we found that in the second independent cohort, many of the same pathways were also significantly altered. And in the progranulin mutation carrier cohort, many of the same pathways were still altered. And it's important to note that this cohort was of 80 patients, this one was of 40, and this one was of 24. And so the power decreases with the lower number of patients. And so I was very pleased just to see that we had so many of the pathways that were significantly changed in the smaller cohorts. Next, so we found that the two independent sporadic frontotemporal dementia patients and the progranulin mutation carriers all had similar changes in the pathways of the protein networks in the blood. And we wanted to see if the progranulin mouse model was also a good model of the changes in the plasma. So at the time we did this, we did not have an antibody array that we could use for mouse proteins, and so we used Luminex, which is a technology that can assay about 60 proteins at the same time. And we found that there were increases in pro-inflammatory cytokines in the mice as well, including TNF-alpha, which is, as I mentioned, was upregulated in the frontotemporal dementia patients. And when we used ingenuity pathway analysis to see the connections between the proteins in the mouse model, we found, again, that there was a network centered on TNF-alpha that was significantly changed in the mouse model. So to summarize this section, we found that in the plasma, we found changes in protein levels that can differentiate frontotemporal dementia patients from controls, that similar networks were altered in two independent sporadic disease cohorts, as well as the progranulin mutation cohort and the progranulin mouse model, and that the IL-12/23 signaling is altered in frontotemporal dementia. And we wanted to see if the changes in the systemic environment were reflected in the brain. And it is known that neuroinflammation is increased in frontotemporal dementia patients. And the most common way to assay neuroinflammation is by looking at microglia, which are the immune cells of the brain. You can see in the swimmer stain that microglia have these beautiful processes that are constantly surveying the environment of the brain, and they are ready to respond to infection or to damaged neurons. And two ways to measure the increase in the microglial activity is using a CD68 stain and an EVA1 stain, which helps us to visualize the activated microglia. So here in the humans, you can see that as TDP43 pathology, which is the underlying protein aggregate, increases, the microgliosis as measured by CD68 and by EVA1 increases, and this correlates well with TDP43 pathology. In the knockout mice, microgliosis is actually the strongest phenotype that the mice have. They have a huge increase in CD68 staining, and you can actually tell very easily just by looking at a slide of the sections which animals are your knockouts and which ones are your wild type. It's difficult to do blinded studies. And it's also interesting to see that we developed a flow cytometry panel to look at additional changes in the microglia of the progranulin mice. And with Ava here, I found that F480, which is another macrophage and microglial activation marker, is highly upregulated in the progranulin knockout mice. Also, it's been found that intrathecal levels of both TNF-alpha and TGF-beta are increased in frontotemporal dementia patients. So we know that neuroinflammation is increased in frontotemporal dementia patients as well as systemic inflammation. And I mentioned the IL-12/23 signaling axis, and we wanted to see, because this is such an important immune regulating pathway, it's likely that the cellular fraction of the blood is also altered. And so I hope to see if there were changes in the immune cells of these patients as well. And we wanted to do this in an as unbiased way as possible. We had a very unbiased way to assay the proteins, and we wanted an unbiased way to look at the immune system. And so we used the gold standard of flow cytometry, but updated a little bit. So for the non-immunologists in the room, the standard of looking at immune cells for the last 20 years has been flow cytometry. And the basic way that this works is that you have antibodies conjugated to fluorophores that bind specifically to receptors or molecules on immune cells, and you can tell by the labeling of the immune cells which cells they are, and you can detect them by the fluorescence. However, we are very limited as to the number of parameters that we can assay per sample because we are limited to the light spectrum, and there's a lot of spectral overlap between the fluorophores. And so we used an updated technology for flow cytometry that was developed largely here at Stanford, and it's called CyTOF, or cytometry by time-of-flight mass spectrometry. And instead of using fluorophores to label the antibodies are labeled with lanthanide series heavy metals. And these are metals that are not found in normal biological sample, and so we know that if they're present, it is because they were on the antibodies we were trying to label. And we can read this with a mass spec and still get single-cell resolution. So instead of using a theoretical maximum of 18 parameters in traditional flow cytometry, we're able to, in theory, use up to 100 parameters. And the chemistry right now enables us to assay about 40. And the advantage of this is that we can get a much broader idea of the changes in the immune system. So for my study, I collected peripheral blood mononuclear cells, or the white blood cells, from frontotemporal dementia patients and controls. And I used 34 surface markers to measure at least 1 million cells per patient, and this was done with the Human Immune Monitoring Center here at Stanford. As you might imagine, using 34 different markers per sample and having 1 million cells per patient gives you a large data set that you need to have new tools to get a good visualization of. So the first analysis that I did on these cells was the SPADE analysis, which is spanning tree progression analysis of densely normalized events. And what this enables us to do is to condense large dimensional data sets into 2 dimensions so that we can get a handle on it. So basically, this works by taking my 34-dimensional data, downsampling it into a more manageable size, then using nearest neighbor clustering to group cells by their similar features, and then uses a spanning tree to link the clusters that are similar to each other. And so we end up with these trees that have bubbles that are representing specific populations of immune cells, and the size of the bubble indicates the number of cells in that population, as well as the color in that case. And the proximity of the clusters to each other indicates the similarity of the cells to each other. So we ran SPADE on all of our patients, and we found some very interesting changes. And I'm showing here a representative plot from a control patient and an FTD patient. And those of you with very good eyes might be able to spot some changes, which I will point out. So the first change was in, that I'll talk about was in CD8 cells, which are cytotoxic T cells. And this population is expressing CD27. So I'm going to zoom in a little bit. And you can see that this patient has a much larger population of CD27 positive CD8 cells than the control. And CD27 is a marker of naive cells on T cells. And it is part of the co-stim receptor that helps the T cells to get the second signal that they need to be activated. So it's usually downregulated after the T cell is activated. And it's also important to note that it's a TNF-alpha family member. And we found, when you look at all of the patients, the frontotemporal dementia patients have a uniformly high percentage of CD8 T cells that express CD27, whereas the control patients have a more heterogeneous percentage. The next population that was changed is also in the CD8 T cells, but this time in the CD85J positive population. So you can see here that the control patient has a huge number of clusters that express CD85J, and this frontotemporal dementia patient has almost no cells in that population. The CD85J is originally characterized on natural killer cells, where it's known to be an inhibitory receptor, and on T cells, it's thought to be a marker of senescent memory cells. So it's known to bind to HLA and act to inhibit the ability of the cells to be activated and to secrete cytokine. And you see a huge increase in the number of CD85J positive cells in chronic viral infections, such as cytomegalovirus infection. And we saw that in the opposite direction of the CD27 population, we found that the FTD patients had a uniformly lower percentage of cells that express CD85J, whereas again the controls were more homogeneous, as you would expect in an aged population. The last change that we saw was in natural killer cells, which are cells that are able to recognize, which are cells that are able to recognize foreign cells and lyse them. So in this case, the change is very subtle, but you can see slightly larger populations of this particular NK cell subset in the frontotemporal dementia patient. And this particular subset is the CD161 expressing natural killer cells. And CD161 is also an inhibitory receptor that sends a signal to the natural killer cell to not lyse the target cell. And in this case, we found that the frontotemporal dementia patients had an overall increase in the percentage of the natural killer cells that are expressing this inhibitory marker. So to summarize, we found changes in the CD27 positive CD8 T cells as well as CD85J positive CD8 T cells. And these populations are probably the inverse populations. So when you gauge CD27 and CD85J together, you find that the populations do not overlap. So the cells that express CD85J do not express CD27. We also found that there was an increase overall in the percentage of natural killer cells that express CD161. And as far as putting this into context of other diseases, I mentioned in the plasma section that there were similarities to autoimmune disease in the plasma signature of the proteins. And we also found that there were similarities to autoimmune disease in the cellular signature of the proteins. So here you can see that CD27 is a target of autoimmune therapy in several diseases. CD85J on T cells has been found to be decreased in lupus, as well as polymorphisms in CD85J are associated with rheumatoid arthritis. And in rheumatoid arthritis as well, there has been found a similar increase in the numbers of CD161 positive natural killer cells. We also wanted to see if the mouse model replicated this phenotype, and the only cell type they were able to assay in the mouse because of the differences in the immune molecules of the mice was the CD27 positive CD8 T cells. And we did find that similar to the humans, the granular knockout mice had a uniformly high percentage of CD8 cells that were expressing CD27. So to summarize this section, in the immune cells, we found that the frontotemporal dementia patients have a high percentage of CD161 positive or inhibitory natural killer cells, and a shift in the CD8 T cells from CD85J positive to CD27 positive. And the mice also had the change in CD27 positive T cells. So the next obvious question was, do the frontotemporal dementia patients have an increased comorbidity of autoimmune disease? And so we asked our collaborators at UCSF who see most of the frontotemporal dementia patients in the area if there was any correlation there. And we asked them to do a retrospective study of the patient histories and note if the patients had autoimmune disease. And we found that indeed there was an increase in autoimmune disease. So this has been published, and we found that in a control population of aged individuals, the expected percentage of autoimmune disease incidence is about 4%, which we found in this cohort. However, in the frontotemporal dementia patients of the sporadic semantic dementia variety, we found that there were 12%, which is a threefold increase. And I'll note here that you're probably wondering what the white bars are, and this is thyroid disease only, which the clinicians said they weren't able to determine if it was from an autoimmune disease cause or not, so they listed it separately. In the progranulin mutation carriers, there was also an increased incidence of comorbidity of autoimmune disease. So you can see that 13% of the patients had autoimmune disease. And it's also important to know if this is a common feature of neurodegenerative disease or not. And so we also looked at a collection of Alzheimer's disease patients, and they found that the Alzheimer's disease patients had identical levels of incidence of autoimmune disease compared to the controls. And very excitingly, there is new evidence that there might be an additional genetic link to autoimmune disease. So my very astute committee a couple of years ago in a committee meeting saw this data, and they said, you need to look in the genome-wide association studies and see if you can find any association with HLA, which is human leukocyte antigen, which is found to be associated with most autoimmune diseases have a linked to HLA. And just last week, there was a study that did GWAS, a genome-wide association study, on a large cohort of frontotemporal dementia patients and found HLA as the top hit. So this indicates that there are also genetic underlying propensities for autoimmune disease in this cohort. So the next question that I wanted to address was, might these pathways offer a new therapeutic target? And we chose TNF-alpha as a potential therapeutic target because we found it to be increased in both the progranulin mutation carriers and the somatic dementia sporadic cases, as seen here in MEF and in the progranulin knockout mouse. And so we used the progranulin knockout mouse and treated them with etanercept, which is an antibody that binds to TNF-alpha and deactivates it. We treated the mice for 3 months, and we used 15-month-old progranulin mice. And 15-month-old mice have already pretty severe pathology, and so we were hoping just to see a small benefit of this treatment. And I would like to thank Ava very much for doing this study with me. And she did this behavior, which is beautiful. And so behavior we assayed in the mice is peer conditioning behavior, and this tests for learning and memory. So this test uses a chamber in which the floor has the ability to deliver a mild electric shock to the mice, which they find unpleasant. And so they should remember when they're put into this chamber again that something rather unpleasant happened there. And the way that mice express fear is by freezing. So we can measure the amount of time that the mice freeze, and this correlates to their ability to remember that they were in this unpleasant chamber. We find a very nice separation of the wild type and the progranulin knockout animals. So you can see here that the wild type animals remember very well that they were in this unpleasant chamber, whereas the progranulin knockout animals don't remember as well. When we treated the wild type animals with etanercept, there was no significant increase in their ability to remember the chamber. But when we treated the progranulin knockout animals with etanercept, there was an increase in their ability to remember the chamber. And this is not significant, but it is an important trend, and it's worth noting that this is only an N of 7 for the progranulin knockout animals. So these studies are ongoing, and we hope to see improved memory in the animals. An additional benefit of etanercept in the progranulin-treated mice was a little unexpected, but very interesting. So in 15-month-old mice, the progranulin knockout mice especially are very likely to die. They're not very healthy, and we lose a lot of them. What we found was that in the study, 3 of the progranulin knockout animals died during the course of the study that were not treated with etanercept, whereas none of the animals that were treated with etanercept died during the course of the study. And clearly this needs to be repeated, but I think it's a really exciting link. Another potential therapeutic target that our data suggests is in JAK-STAT signaling. So JAK-STAT signaling is a common signaling of many of the molecules that we found changed in the disease, and there are already FDA-approved drugs on the market for this. And so our collaborators in a consortium for frontotemporal dementia research are very interested in trying to get this into a clinical trial. So to summarize, we found that in the frontotemporal dementia patients, both the sporadic and progranulin mutation carriers and the mouse model of progranulin have an increase in systemic inflammation that can be observed both in the plasma proteome and the cytokine signaling and in cellular immunity. There are important links to autoimmunity from both the cellular side and the protein side, and these might offer new therapeutic targets for the disease. I would like to thank the Weiss Gray Lab, who's here in full force. As you can see, we are growing, as well as my Stanford collaborators in the Nolan Lab and the ACE Lab and the Human Immune Monitoring Center. And my wonderful committee who always has great ideas, Phil and Emma Marion, who was unfortunately unable to be here today. And so thank you to Kathy for stepping in. And thank you very much to Pat for agreeing to be the chair of my committee. I'd also like to thank my funding sources and my outside collaborators, including many at UCSF. And with that, I'm happy to take questions and stick around for some additional acknowledgments at the end. Yes. Oh, are you asking which autoimmune diseases I've seen in the patients? It's actually a huge variety of different autoimmune diseases. So there wasn't any particular disease that really stood out as being the primary association with frontotemporal dementia. There were certainly a few with arthritis. There was a couple with Sjogren's. There was some with skin autoimmune disease-related conditions, but no particular autoimmune disease was seen to be much more highly associated with frontotemporal dementia than the others. Does that answer your question? Yes?
Did you see changes in total white blood cell counts, or was it
No.
change in lineage?
Only changes in lineage. So the total count was unchanged, as well as the total percentages of, say, CD4 and CD8 cells.
So were those cytokines involved in those cells? You took the proteins from that plasma and applied it to those cells.
I did that. Let's see if I have those slides.
You don't have to be talking about it.
Sure.
Yeah.
What we found was, I have a lot of extra slides.